Sharks and rays. They are often dismissed as monsters of the sea. But these majestic, enigmatic creatures are actually essential wardens of our marine environment. Found in every ocean across the world, they play a fundamental role in ensuring balance and stability in our oceans. Sadly, this role is jeopardized by overfishing and habitat loss. Around 3 billion people rely on fish as their primary source of protein. Ongoing declines in shark populations could have devastating and unpredictable impacts on marine food webs and even our own food supply. The plight of sharks and rays was the spark for the Save Our Seas Foundation, founded over 15 years ago. Through the funding and support of research, conservation, and education initiatives, we are dedicated to safeguarding the future of sharks and rays. Today we have 3 centers based in Seychelles, South Africa, and the USA. Since 2003, we have funded over 300 projects on over 60 species in over 50 countries. We have also partnered long-term with several projects, including the Shark Spotters in South Africa, the Shark Lab in the Bahamas, and the Manta Trust. We're passionate about our mission: protect the future of sharks and rays and our oceans for generations to come. We can and will make a difference. The Save Our Seas Foundation. Sharks and rays. They are ancient, enigmatic, misunderstood, and essential to our oceans. But these important animals are under immense pressure. As the Save Our Seas Foundation, we're here to help. Through supporting research, conservation, and education, we work to ensure a future for sharks and rays. The Save Our Seas Foundation. Together, we can make a difference.
Hello, everyone, and welcome to the Save Our Seas Foundation Distinguished Speaker Series, hosted by the Museum of Discovery and Science. We are so happy to see you all here tonight for our discussion on deep water sharks. We have two wonderful experts, all that have all this wonderful knowledge about deep water sharks joining us this evening. I'll introduce them in one moment. My name is Lance. I am the Director of Environmental Sustainability here at the Museum of Discovery and Science. So we do a lot of really cool things here at the museum involving environmental sustainability. Our main goal is to connect people to inspiring science. So if you've ever visited Fort Lauderdale, please come by, stop by our museum. It's a wonderful place for children from 0 to 99 and beyond. So joining us this evening, like I said, we have two experts. We're lucky enough to have two deep water shark experts from the Save Our Seas Foundation, and to talk all about their projects and what they have, and all the amazing things that they've done and discovered and what they plan to do. So they'll be here to share what they're up to. First, we have Sofia Graça Aranha. Sophia is a PhD candidate in environmental and marine sciences and the project leader with Elasmop, a project that aims to assess the condition and survival of elasmobranchs, sharks and rays, caught in fishing operations in the Northeast Atlantic Ocean. Sophia grew up in Rio de Janeiro, and as a child, she used to watch Jacques Cousteau documentaries with her grandmother, and remember being very, very drawn to the ocean. Sophia followed that curiosity and decided to dedicate her studies to sharks, the most feared, loved, and important animals of the ocean. As part of her postgraduate studies in Brazil, she developed a shark conservation project called Shark Free, aiming at indirectly impacting the shark-targeted fisheries. During her master's studies, she assessed the nutritional status of deep water shark species on the southwest coast of Portugal, using little explored methodology involving DNA and RNA, and looking at their diets using isotope analysis. Currently, she's a doctoral student. Sophia is continuing the same work developed in her master's degree, expanding the focus to deep water sharks, skates, and chimeras, and evaluating their survival rates for crustacean bottom trawl activities near Portugal. As part of her PhD project, the project Elasmop is contributing to the understanding the factors affecting survival of elasmobranchs, and is expected to propose new fishing procedures and handling for these animals on board commercial fishing vessels to diminish their bycatch and increase survivability. So, sounds amazing. Welcome, Sophia. Glad to have you.
Thank you, Lance. Thank you for the introduction, and thank you for having me, for inviting me here over. It's a pleasure.
Yes. And like I said, we have two wonderful experts tonight. Our second is Dr. Diana Catarino. Diana, for as long as she can remember, Diana has had a passion for nature and its inhabitants, especially for the sea in particular. She grew up in a small coastal village close to Sintra in Portugal, and spent summer holidays playing at the beach, as we are lucky to do here in South Florida. She was always very curious about creatures in the rock pools, and would dedicate part of her day to checking what the tide had left on the sand and on the rocks in the pools. Still a thing that she does today when she goes to the beach to check out those tide pools. While snorkeling on her trip to the Azores, Diana fell in love with the islands and the amazing display of colorful fish underwater, as well as the rich biodiversity and huge schools of fish. Diana's career in the ocean started with her work on deep sea demersal surveys at the Department of Oceanography and Fisheries at the University of the Azores. This is where her passion for taxonomy, ecology, and conservation of deep water sharks and rays began. In 2018, she completed her PhD, congratulations, doctor, in marine sciences, studying the ecology and population genetics of deep sea fish and sharks, and furthering her knowledge about this poorly known ecosystem and all the threats that face it. Diana's current research interests combine state-of-the-art molecular tools and deep water shark and ray taxonomy, and how they can be used to help protect this diverse diversity of these species. After all these years, Diana is still driven by the same curiosity she has as a child, wanting to know more about these stunning and mysterious animals and to promote their conservation so that they will continue to amaze future generations. So, Diana, thank you so much for joining. Welcome, welcome.
Thank you very much for having me. It's a pleasure to share a bit of what I do with other people.
Yeah, exactly. Well, we're happy that you're here to share. Just a few things for all of our guests logged in. If you have questions, please feel free to put them in the chat. I'll be monitoring the chat as we go through. Sophia and Diana both have a little overview that they're going to share with us of their projects that they've conducted with the Save Our Seas Foundation. If you have a question while they're presenting, and please feel free to type it in the chat. We want to make this an informal conversational interview, so don't be shy if you have a question. So we'll give time to Diana and Sophia to explain their projects, and then again we'll have some dedicated time at the end for questions as well. So we flipped a coin a few days ago, and Diana was lucky enough to go first. So Diana, could you please give us an overview? What is your project funded by the Save Our Seas Foundation? What have you been up to?
Yeah, I can start sharing my screen.
Yeah, please do.
Yes. Can you see it?
You can.
Okay, right. Sorry. Oh,
there you go. Perfect.
Yeah. So hello, everybody. I'm Diana, and my project with the Save Our Seas, it calls Sustainable Science in the Deep Blue Sea. And this was done also in collaboration with another project called SOS Tubac-Pro, and also with the demersal surveys in the Azores, to go on board and study a bit of the deep water ecosystems and the deep sea sharks that live on it. So first, maybe for some of you, the Azores are not very well known, so I leave you here a bit of where the Azores are. So basically, we are in the middle of the ocean. You can barely see us there, but we are there, and more or less halfway between Europe and America. And basically, we are 9 volcanic islands and several seamounts that sit right on top of the mid-Atlantic ridge. And the exclusive economic zone of the Azores has on average a depth of 3,000 meters. So it's quite deep, and we have literally the deep sea right around the corner. So my institute, OKEANOS, has developed a lot of research in the deep sea because it's so easy to access to it from here, from where we live. So a bit of background on deep water sharks or elasmobranchs. So we basically consider deep water elasmobranchs those that live at more than 200 meters deep, or that spend most of their lives at that depth. And they are considered quite vulnerable species. And why is this? Because they have like life history traits that have particular traits that makes them quite vulnerable to any disturbance that in their ecosystem. So they are very long-lived living species. So some of them can reach 50, 70 years old. And one of the extreme cases is the Greenland shark that you may heard of, that can grow or can reach 400 years old. So they grow very, very slow. They also mature quite late. Some take like 15, 20 years to mature. The Greenland shark, again, extreme, 150 years to get first maturation. So for most of them, also they have very few pups. So many of them less than 30 pups per liter, and some of them even only 1 or 2, which is quite low. And then if you think that some of them have pregnancies that long up to 3 years, then the generation time to renew population is quite long. So for the in the Azores, we have basically that we know about 25 species of deep water sharks, and plus 5 skates or deep water rays. They are listed, or most of them are listed in the red list by UCN, which is the species that assess the vulnerability of the species worldwide, and half of them are either with some level of threat, or just are considered data deficient, which means that we don't have enough data to know what's the status of the species for those. And if you want to know a bit more about the deep water sharks that we have in the Azores, you can check out this deep sea sharks guide that we have for the species that occur in the Azores. If you know a little bit of Portuguese or maybe Spanish, or you can always try to Google translate it. It has some interesting facts for the 25 species that we have here around the islands. Yeah, okay. So why we need to monitoring deep water sharks? Why is that important? Well, mainly deep water sharks are common bycatch of the commercial fisheries. In Europe now, they are considered prohibited species, which means that if a fisherman catch a shark, they have to return it to the sea. But then we don't, because they are vulnerable species, we don't know actually if they are surviving once they return to the sea. We don't know what happens, and because they are forbidden, so they cannot bring them to land, then we also don't know how many are actually being bycaught. Because of their life history traits, they are very, very vulnerable to overfishing, and we don't know even if they are vulnerable to small amounts of bycatch. So actually, it's very important that we monitor this species to just ensure that conservation and ensure the sustainability of deep water fisheries in general. So by monitoring, what do we mean? Usually we have like this traditional way of monitoring species, which is basically go fishing for them or perform fishing surveys, scientific fishing surveys. Here in the Azores, we use long lines to do that. So basically, you go and see how many fish and deep water sharks you catch, and try to have abundance for those deep water sharks. But in the end, fishing surveys are quite invasive and potentially lethal, because you have to fish for them and they come from great depth. So when they return to the water, they are usually not very well. So it will be quite interesting, because I mean, this is vulnerable species that we have other ways to get these assessments for fisheries and for stocks, and to have other ways to get the same data. So what I'm going to do is trying to have non-invasive methods to do it. So basically, there's two non-invasive methods that I choose, and one of them is BRUVs, or beta remote underwater videos, and basically just you send some cameras down with the bait, and you film what comes to the bait, and you try to infer abundance from there and see which species will come to the bait. Another method is environmental DNA, which basically is based in water samples. You just take samples from the water in the environment that you want to research or investigate, and you infer species community from there. So just for those that are not much familiar with the environmental DNA, it's just basically when some fish or shark go through the water, it will shed some DNA into the water by the skin, by the waste it just released in the water. And from this, you can collect, in theory, a bottle of water, and then investigate what DNA is on it. And because of DNA barcoding, which basically is the assumption that you have a gene that has a specific sequence for each species of fish or shark. So is that a species of shark has a specific sequence, another shark, another sequence, then you can tell them apart just by the DNA. So what you're looking for, when you look this in the water, you are just looking for a meta barcoding approach, which means that you're looking for all the sequence from different species in the water at the same time. And then you can infer which species are there just by the DNA that they leave in the water when they pass by.
That's really amazing.
Yeah.
Just by taking a water sample, you can see what was there. Like how long? I just have just a quick question. Sorry to interrupt, but
no problem.
How long does that DNA stay in the water, like intact enough for you to like determine what species is there?
There's still some doubts about how long it can take. There's some studies that indicate 24 or more, or even more time. It also really depends on the kind of microbial activity of the area, also the temperature, of course. And in the deep sea, we actually don't know. And then, of course, currents, because I mean, if the DNA is there, if there's a strong currents, it can just be swept away to other place.
Right.
So in the deep sea, we don't really know how long, but at least maybe a few days.
Yeah, that's really interesting.
And
that's
also,
cool.
If it's fresher, then you have more chance to amplify it. So, like fresher DNA in the water will be amplified easier than like more degraded one.
Right. Thanks.
So about the objective or the aims of this work, basically, the main thing that I wanted to do is just to compare the efficiency of non-invasive methods like the eDNA and the BRUVs in assessing the deep sea elasmobranch diversity and relative abundances and compared with the traditional methods, which basically is long line fishing, and see how well they will perform. And of course, we also want to collect new information about the spatial deep water elasmobranch biodiversity and relative abundance across the Azores. And also will be quite interesting to compare this abundance in protected and non-protected areas and try to see if we can get a signal there. And of course, all of this data together will be amazing to identify essential habitats, which basically is habitats that are crucial for several steps on the life of the sharks, like nursery areas, aggregation areas, and also species hotspots, because then it will be quite interesting for conservation to have those areas identified. So just a bit to show you overview what I've been doing. So for the fishing service, which is a long line or bottom long line surveys. We've been on this annual demersal surveys. We perform a total of 56 bottom long line deployments from the coastline to 1,200 meters down. And then we use metrics for calculate catch per unit of effort. So for the eDNA metabarcoding, then we use 56 water samples at the maximum depth of the fishing set, which was, we collect water basically between 600 and 1,200 meters. Then there's a whole protocol that we use for filtration of the water, extract the DNA. We use the 12S region, which is a mitochondrial DNA gene. And then we do the metabarcoding. And eventually with all the filtrations, we need to match the DNA that we get in our sequences to online databases that are reference for the species that we might encounter. And this is kind of a crucial step. And then for the BRUVs, unfortunately, we only had the chance to do 6 deployments, which is quite low. And we did it at the same time as the fishing sets during 2021. And for this, we use a structure adapted from the Azores drift cam. It basically is like a metal apparatus that has a very potent light and a camera and a bait. And then we send it down to between 700 and 800 meters and record for a very, very long time. So about the results that I obtained. So regarding biodiversity, basically, we could identify the total of 23 Elasmobranch taxa. Some of them could be identified at species level, some of them mostly with eDNA to the family or genus level. So with the bottom long lines, we identified a total of 13 species, with eDNA 10 species, but then also plus 8 to the genus or family, and only 5 species with BRUVs. So the thing is that that you told you about the online reference database for sequence or for eDNA, then the thing is that you really need to have a reference sequence in the database to know which species is that. So when we don't have a match to 99%, then we get a family or a genus. And then what we found here with eDNA is that actually the species that only got identified with the bottom long lines are those that are not in the database. So there's no genetic reference for any of these species here on the bottom. So when this reference is done, probably what we'll see is they are here in these families. And this also with the eDNA, we saw that there's a lot of matches that at different percentage of identity. So maybe there's also some room for new records or cryptid species. So in total, basically, from the 30 deep water elasmobranchs that we have in the Azores, with the 3 methods, we could identify or detect 15 of them. About the relative abundance, which means like which species are more abundant than the others. So, and here we see that eDNA actually gets quite a great potential of detection. We see that we could correlate the number of reads that we found with eDNA with the CPUA or the catch per unit of effort from the log lines, which means that more individuals that we get with the long lines, we see the same pattern with the eDNA, and there's some correlation in there. So this seems great potential for having eDNA as an alternative or to long lines, or even a complementarity to the long line methods. And also with the improvement of the online databases, it's possible that this correlation will improve, because some of the species that are not in the reference genetic database are some of the most abundant. So these are not in here. Only here are only the species that were identified to the species level and are in both eDNA and long line. So the ones that only to the genus or family level were removed from this correlation. So there's some room for improvement here. So about the proofs. The proofs, unfortunately, we cannot do any relative abundance calculations and compare with the other methods just because we have too few sample sites. And what we saw is that for the proofs, the first arrival time for the elasmobranchs is actually near 3 hours, which means that we have to record for a very long time to get any elasmobranchs. And still, we have very few species appearing and also few individuals. So for a normal groove in the shallow water that usually you do it or you record for one time and you still have abundance, for the deep sea is much more challenging because usually animals are more far apart from each other, basically. But there was some surprise with the grooves. Actually, one of the most abundant record that we had was the flopper skate, which is a critically endangered species, and that we only catch a few times with the long line. So this was quite a good surprise. And also with grooves, the good thing is you get really cool videos that we can use for outreach and other things. And also it's very nice to see the habitat where the species are. So if you increase sampling size, then you can get some patterns of habitat preference. And also, if you have lucky, you can also determine some essential habitats. And about 3 weeks ago, we got very, very lucky because we found a deep sea shark nursery area. So we were very excited about this. This was an area we found probably 1,000 of eggs, or maybe like the hundreds and hundreds of eggs. And we're still trying to investigate a little bit. And but yeah, this is a really nice result because deep sea essential habitats for deep water sharks are quite difficult to find. So we're very excited about it.
Those are egg cases they're out there.
Egg cases, yeah.
Wow. Okay.
Very cool.
Do you know the species? Sorry. Do you know the species, Diana, or you're still investigating?
We're still investigating.
Okay. Cool.
Yeah, this was quite recent.
Yeah.
So about the spatial analysis, well, I still didn't have much time to get really into it, but what we really want to do is kind of compare eDNA results with long line in a spatial scale. And what I show you here is just like a really preliminary thing that I did that really shows that might be, for instance, some difference between islands and seamounts in for deep water sharks eDNA data. So, and this is something that we expect with the fishing data. So it's quite nice to see that it might be some similar pattern there, and then that we can really correlate both methods, and they are basically giving the same results. So, but I still need to investigate this much further, and also I really want to compare MPAs and no MPAs data, because it will be very cool to see an eDNA signature for an area that has been closed for some time, so probably has more fish, and also will have a signal for the predators with higher abundance or even biodiversity. So to just in summary, give you a bit of overview of what have been done, or the main results. So eDNA metabarconing is quite promising monitoring tool for deep water elasmobranchs, and we seem to have some comparable results to the long line catches. Also, it shows potential as a complement or alternative non-invasive method to the traditional sampling, both for biodiversity and also abundance studies. But of course, detection efficiency can be improved, of course. just by getting better online reference databases. And this is quite easy to do. We just have the tissues from the sharks, and then we just have to sequence for those genes and then put it online. The BRUVs reveal some potential for biodiversity. Of course, with increased sampling size, this will be quite great to see different species in different habitats. But from our perspective, they are a bit limited efficiency for relative abundance, just because we have to record for a very long time and only see a few individuals from few species. Nevertheless, with BRUVs, we can see the 6 gills, and it's quite abundant at the flapper scale. So these are species that are not commonly captured with long lines. So this work kind of says that actually a multidisciplinary approach with monitoring is actually the best choice, because some methods are better for some species while other methods are better for others. So if we have different monitoring approaches, then we can have a better overview for different deep sea elasmobranch species. And yeah, basically that was it. So I really like to thank to Save Our Seas for supporting the work, and all the other funders. And if you want to follow the news of this work, and also from SOS to the Prof, you can follow us on social media. Thank you very much.
Thank you very much. It sounds like you're doing some really amazing work. We had a question from the audience about, and then we'll get to Sophia. We had a question about improving the genetic database so as you collect species through the deep water longlining, can you then take genetic samples from those species to improve the database, right? Or the genetic database, so the environmental sampling can be used more, right?
Yeah, that's true, and that's actually is our next step, because we collect a lot of these sharks and also other deep water species a lot. So actually, OKEANOS has a total of, it's more than 22,000 tissues for genetic samples stored. So the next step is actually try to use these samples and complete online genetic database that actually for the deep sea. That's the thing for the deep sea, because there's not many people working with these animals, then the genetic database are always a bit more scarce.
Right. Yeah, it's such a powerful tool. I mean, when I was in high school, I never thought that you could, when I was studying genetics and learning about it in college, even that you could sample something from the environment and then be able to say, oh, look, that species swam by, 24 hours ago or whatever it might be. But it's a really, really amazing tool. And so I know you're doing amazing work to figure out what is there, what's the abundance of these deep water sharks. And some of them long lived, 400 years, 150 years before they reach maturity. That's just mind blowing. It's amazing.
Yeah, and
and I know that's sorry.
No, that's also why they sometimes are quite threatened,
right?
Because I mean, it's a long time to reproduce for the first time, to just
which is why I think Sophia's work is really, really important as well. So Sophia, you are doing a lot of work to improve the survivability of these deep water creatures whenever they're accidentally caught, right? Through commercial fishing operations. How do we lessen that catch, and when they do get caught, how do we put them back in the ocean safely so they can keep going for another 400 years, whatever it might be, right?
I'm trying to figure out what are the threats first, and then to try to find solutions to diminish the threats and how they can be safely returned to the water. But I will talk about it now.
Yeah, if you'd like to go ahead and pop up your presentation, we'd love to hear about it.
Let's do it.
And for those of you that are on, since we do have 2 speakers tonight, we were probably going to go a little bit after 7, because there's already some questions coming in. So I want to make sure we have time to get to your questions. So try to stick around a few minutes after 7 o'clock, and we'll keep going up for another maybe 5 or 10 minutes afterwards.
Thank you, Lance. So I will talk about one part of my PhD work, which is the project DelasMop. The project DelasMop and the other project I will just briefly speak about. They are projects that I had to fund my PhD work, because all I wanted to do was super expensive and I needed money. So I created, me and my supervisor, of course, we created those 2 projects, which I will talk about. DelasMop is funded by mainly by Save Our Seas Foundation, but is also funded by the EEA grants, Portugal. My supervisors are Ivone Figueiredo from the Portuguese Institute for the Sea and the Atmosphere, Alexandra Teodosio from CCMAR and the University of Algarve, and Esther Dias from CIMAR, from the University of Porto. We also have the support, the technical support of OLSPS, which I will briefly talk about. So my PhD study is about the condition, what I wanted to know more, because I started my master's with the deep sea shark. And then I saw that was a huge gap in knowledge. And then I was like, I want to go further for my PhD. So I decided to create a project to evaluate the condition and the survival rate of the most commonly caught deep sea sharks. But first I needed to find out which sharks were the most commonly caught, right? So we created the DelasMop project. DelasMop stands for Deep Sea Elasmobranchs of Portugal, with funding from Save Our Seas. And the other project, which is a little bit different, is about the electronic monitoring and reporting for fisheries in Portugal, for bottom trawl fisheries, which is funded by EEA grants. And we have those partners which are mentioned here. And we use my PhD study as a case study for this MREP project. So the motivation of my work is mainly what Diana just talked about. So I will not get into much details here because you all know about deep sea sharks and skates, and I will not bother you much with that. But those type of studies, ecophysiological studies, they are really important for the management of fisheries, right? We want to know what they are caught, what they are catching, why they are catching, how can we diminish that, and why is that important, right? So why in the south of Portugal, which is where I'm developing my work, is because it's the zone with the highest fishing effort for crustacean bottom trawlers in Portugal. So the south and southwest coast contains a lot of bottom trawlers, and the deep water sharks and skates, they are heavily caught by this fishery. As Diana already said, they are meso to top predators and are highly susceptible to overfishing because of their life histories. They mature late, they grow slow, and they have a few offsprings, mainly because of the high pressure and the low temperatures with depth that diminish their metabolism. So they are more susceptible because they are meso to top predators. They are more susceptible to overfishing. And also, because very little is known about the deep water elasmobranchs and their interaction with fisheries, we created this project. The main questions we want to ask with DelasMop is which are the main species that are caught by bottom trawlers in southern Portugal? If they survive this fishing, even when they arrive on board and even after they are discarded, and what influences their survival rates, and what do they eat? Because if they are eating shrimps, they will have a higher chance of being caught. If they prefer commercial shrimps, they will have very high chances of being caught. So this is a bottom trawler. It's a boat with a net that simply swap the bottom of the seafloor. They normally go from 200 meters depth, which is the depth of the deep sea, where starts the deep sea, for until 800 meters depth in Europe, which is the legislation. Bottom trawlers cannot go deeper than 800 meters depth in Europe. So the work was conducted in two vessels. One was the research vessel from Mário Ruivo from the Portuguese Institute for the Sea and Atmosphere. We developed two campaigns there with more or less of 54 days, 51 hours of effort, and 13 kilometers square area was swapped. The depth was from 27 to 776 meters. That was the depth where we found deep water sharks and skates. So commercial crustacean bottom trawler, we conducted 10 campaigns with more or less 33 days in total, 351 hours of fishing effort, 95 kilometers square, and we fished from 96 meters to 1,244 meters depth. I know it's not allowed, not my fault. Okay, so here is the density area, is the density map and the study area. We are in Portugal, as you can see in this square. This is the area where we work. This is the south and the southwest, and those little squares are holes where we collected deep water sharks and skates. They were generated with the software ORAC DDM from the company OLSPS, and the redder the square, more sharks and skates. But the squares, the red squares can go from 40 to 479 individuals. Okay, so we can see where they are more or less, where we have more sharks and skates in this area. So this software was developed for this project and the other project. And here are the species, some of the species that we collected for both projects. In total, they were 21 species, 2,985 individuals, 16 species of sharks, and 5 species of skates. These are the most, this one is the most common shark, the black mouth cat shark, Galeos melanstomus.
It looks like there's some overlap between Diana's species and the species there from Portugal too, right?
Yes, yes. But also she has some species that I never found during the surveys we did. But for example, Diana has found Isancus griseus, which is the 6-gill shark, if I'm not mistaken. Her Dipturus, I have Dipturus nidarusiensis and Oxyrhynchus, and hers is intermedia. I never found intermedia here. So there are some species that I don't know, but I don't know if Diana found them, but it was just not mentioned in her presentation.
Yeah, of course.
But there are some very rare species. For example, the goblin shark.
The goblin.
It's a very cool shark. We only found it once, and it was when the trawler was fishing in illegal depths. So goblin sharks, field sharks.
I didn't know that they were endemic to the that side of the Atlantic. That's really amazing. Yeah. Yeah.
I don't think they're endemic here, but they also occur in Japan.
Right.
In Japan, they also occur in other parts of the world. So, but it's a very uncommon, very unusual shark.
Right.
I have a video of it. I can show you later.
Yeah.
It was really sad. He was barely alive when he got on board. So but then from this shark, it died during the procedures. And then we collect the head, we clean the jaws and the snouts, and we are using for the environmental education that we are doing for the Save Our Seas project. I will talk about it in a bit. So during these cruises, during this service, we collect information about, we identify the species and we put into the software, the ORAC DDL, where we logged all the information here. We collect information on the species. We see if it's a male or female. And here you can tell if it's a male or female. I will let you guess what you think, what I'm holding in my hands. So, so this is a male shark.
Let us guess. That's fine.
Sorry, I ruined it. Okay, so this is Pedro. He's our intern. He's weighing the shark. We only weigh sharks that were dead because it's a very intrusive weighing method. We measure them, and also for the other project, we were collecting blood samples for the live animals. We also, when they arrive on board, we put them in this tank here. Here I'm holding a field shark, and there are other sharks that, trust me, they are alive, but they are barely breathing and they are barely moving. So that's why I have my questions if they will survive after they are being returned, because due to the lack of pressure, they get buoyancy. They buoy. They like a buoy. So
right, they're used to being in the deep ocean, and when they're hauled up to the shallows
exactly.
and out,
and then in 20 minutes they are up. Yeah.
Yeah.
Who knows what this can cause them. So this is not a deep sea species, this one that I'm holding, but for another project, I'm also conducting survival studies in which we collect these species, some commercial skate species. We put them in a tank and take them to land and see if they survive. If they survive, we put them back in the water. And if they do not survive, we open them and see if this might be related with the fishing methodologies. So for the live sharks, which are bigger than 30 centimeters, we also put this tag, but unfortunately, we didn't have too many live sharks that were in good conditions to put a tag and being released. So this is a tag and recapture tag, a plastic tag. We put this tag in over 200 animals, and we didn't have any news about any of them. So either they do not survive, either they are hiding, I don't know. But anyways, this tag is plastic tag. It doesn't hurt the animal very much. We put in the flesh of the animal. It doesn't get, they barely feel it. So it's less intrusive, and it's good if they are recaptured. We know if they survive and how long they survived after they are released. So, and then we share this poster in the fishing ports to know if a fisherman collects it, that they give us the information. So with the dead animals, when they arrive dead on board, we open them. Here I'm holding an egg capsule of Dipterus nidarusiensis. I will show you. They are golden capsules. They're gorgeous. She was dead. Most of those kids arrived dead on board. We see what they eat, what they were eating. We take notes. We also collect muscle samples. And with those muscle samples, what we do, we put them in these plastic flasks and then take them to the lab and evaluate if we evaluate RNA and DNA ratios to know if they have been feeding and in the past few days. That, along with some morphometric measurements, will give us some insights into their feeding ecology. And also we collect muscle samples to evaluate the isotopic signatures to know what possible prey they are eating. And for that, we also need to collect muscle samples from their possible prey. So lots of work, but it's in the end, it's worth it. We had some rescues also during the way. These are Ciliohinus panicula, which is a type of cat shark. They are not deep sea species. This is Sharkira feeding in the tank. This was Sharkira egg, and you can see that it had the yolk, but it was full of air. And what we did, we collected 2 eggs at this day. We placed them in the water and filled them with seawater in this small aquarium. She developed beautifully. And unfortunately, she ran away from this aquarium. She escaped. And she went to a coastal lagoon. I was super devastated because I was getting super attached to her. And I was planning to release her once she was a bit bigger, but I couldn't do it. And then her sister, Tofu, born a few days, a few weeks after, super cute. Then we secluded the aquarium so that would not happen again. And then in that video at the start, when I'm holding like a shark, it's Tofu. She was released back into the ocean, and it's my one of the greatest things I did.
They're so cute.
Yes, they're super cute. They're super cute and super tiny.
You said Sharkira and Tofu?
Sharkira, yeah.
Yeah.
It was not me. It was we did like a survey in the Instagram, and people just voted, and one suggested Sharkira, and it was awesome.
Sharkira, that's good. Good, I like it.
Super good. So for the Delasmall project, we do environmental educational activities. We go to schools. We also go to center. We have this partnership with Centro de Ciência Viva do Algarve, where we take kids from schools also to the Centro de Ciência Viva. We show them what is a shark. We put the shark skin for them to touch, shark teeth, and all of that. These are miniature shark and skate miniature that was done by Luis Diem, a great artist, a very young German artist that did all the illustrations from my presentation, and also gifted us these amazing miniatures that we put like in an aquarium. Kids love it. And this is the jaw and the nostril of the goblin shark that is being portrayed. We also do fisherman interviews. I have a student that she did that for her thesis, and we are still in the process of evaluating the results. With that and all the information we collected about the survival rates and everything, we are constructing a protocol with better handling techniques and suggestions for the fishermen to release the shark rapidly to the water based on our findings and our results from the project. Because we did see that the deeper they go, more sharks are dead, and many other problems happen. I don't know if someone wants to say something. There is a raised hand.
Yeah, I see that. So if make sure if you have a question, please type it into the chat. We're gonna so Sophia hopefully can wrap up in about 5 or so minutes, and then we'll get to some
sorry, I will run. Okay. So
we'll get to some of your questions. There's a lot of them rolling in.
sorry. Yeah, so just a quick review. In the research surveys, the hauls duration were 30 minutes. And in the commercial hauls, the duration goes from 3 hours to up to 8 hours and 9 hours. So as you can see here, in the research, we have many more sharks alive than in the commercial, right? And then the same goes for the dead sharks. Many more sharks dead in commercial than in the research. And we see that this has to do with whole duration, the depth and water temperature differences that might influence those death rates. So I will quickly run through the main, one of the most interesting sharks that we collected during our study. This is a basking shark. This is a video that one of the fishermen sent to me, is the boat that I work with. It's a huge basking shark, so they are in the process of releasing the shark. So that's not a demersal shark. They go to the water column, but they also go deep, very deep, and they live during the winter in deep waters. And there are some researchers that think they should be classified as deep water shark. Okay, so the sailfin rough shark, Oxynotus paradoxus, is a very intriguing shark. Very little is known about them. But in our study, we saw that the depth range that was already discovered was 720 meters. But in our study, when we fished deeper, 1,200 meters depth, we saw one shark, one of those sharks. So we are presenting a short communication on that very, very soon. So this is the Norwegian skate, the golden eggs that I told you about. They're huge, and I'm holding them here. The six-gill shark, to me, is one of the cutest deep water sharks. It looks like a cartoon. Super friendly face. The goblin shark, it was a female with 2.5 meters size, but it can reach up to 6 meters total length. And this is the frilled shark that we released. Actually, we put it in the tank, and then we proceed and we release the shark. We don't know if it survived, but we hope it did. Okay, the Portuguese dogfish shark is one of the sharks that is the one that can go the most deep that we are aware of. And in our study, we also saw the largest specimen ever found. So another short communication on that. And the kite fin shark, also lovely with the cartoon eyes. That's it. Thank you. We are still processing the results. We are still in the process. I did not go into much details on the results. I just wanted to show nice, for now, nice pictures, and then more is to come.
Fantastic. We'll have to have you back.
Yeah.
Report the results, right? And how we can, what can we do to lower the bycatch of these creatures and increase their survivability? It's a really amazing that you were able to help some of them, right, and get them back into the ocean and hopefully, go on to live a happy life. But so we have a few questions from the audience that have come in while you both were speaking. So we'll go to one, one that's kind of broad for both of you. Both of you had talked about species that were endangered or critically endangered, but at the same time, we know so little about these species and where they live and their distribution. So how do we go through and how do we calculate? Rachel asked this question. How do we know if they're in danger? What tool do we use to calculate if they're going extinct or not? How do we know? Which one of you would like to take a stab at that question first? Sophia?
I can start, and then Diana can complement if she wants.
Yeah, that'd be great.
Actually, these are years of data from, there are some biological sampling activities being conducted. So they are to know the state of the resources, basically the commercial resources, right? And also the information from onboard observers. So all this information goes to a database. They are published in articles by other scientists, and eventually IUCN experts, they just collect all of that, and they just measure if this species is declining or not. But there are some species, for example, the sailfin rough shark is data deficient. And as Diana showed that in her presentation, more than 20% of the species, the deepwater species, are data deficient. That means that we don't have enough information to access their status.
Right.
So,
Dan, anything to add to that?
Yeah, that's exactly that. And because many of these deep water sharks are caught so few times, then that's why you don't know much about it, and there's no trends. And all these assessments are based on the trends and also biological traits, and that's what the UCN experts base their assessments in. And so the thing with the data deficiency is that we actually don't really know what's happening, because they could be bycaught and just not reported, and
right.
There's no information, and so it's kind of a gray area.
Well, maybe your research, particularly looking at environmental DNA, to maybe that could lead to some better assessments, right, of these species, because you don't actually have to see the shark.
Yeah.
You can just analyze the water and know it was there, right?
Yeah,
so I guess, if you can have a baseline for a certain species based on environmental DNA, and then it goes down over time, right? Then it's in decline. Or
that's what we were hoping, that have some alternatives, but because also fishing surveys, of course, they are not perfect. And have several methods to infer this, the status of the populations will be really good for the assessments. Yeah.
And one of the things, I mean, you can kind of think about these species as being so easily harmed, right? Because of their natural history. Maybe we should just protect them all, whether or not we know if they're endangered, critically endangered, threatened or not, right? Is there any regulation? You guys are both based in Portugal, technically. Is there any regulations from the EU that protects these species?
Actually, was more or less what European Union did, because they started to have these TAC zero total allowed catches that were like zero for these deep water sharks, or some of them, not all of them. And now they turn into prohibit species, which means that if you catch one, then you have to put it back or release it to the water. But then the thing is that then scientists also don't get the data, how many are being caught, and you don't really know how many will survive, because as Sophia said, and we also observed this in the Azores, we have a huge problem to make them sink, because they don't sink, they just go belly up in the water, and they don't have strength to go down again.
Right. Yeah, it's interesting. Well, that kind of leads us to another question from a person that's listening in. What can when we think about these species and how the general public can benefit, right? Or not benefit, but like, how can we help the sharks benefit, right? How can we get involved? What can we do as a general member of the public to protect these species? Because, as you were saying, they're top predators. They're critically important to their environment that they live in. And just based on Sophia's pictures, I mean, you can see the bycatch is just crazy. So what can we do? The question specifically is, well, how can we look at fish consumption with humans eating fish from the ocean? Is farming better? But also, you know, what are your opinions on how can we have an impact to protect these species? Diana, you would like to go first?
Yes, sure. Well, especially about the deep water sharks, I guess, well, they already prohibit. They go back to the sea. Then we should try to figure out what is actually the survival rate. But one of the possibilities will maybe try to have bycatch mitigation measures, or eventually some gear selectivity if it's not possible. We have been doing some studies in the other project that we have with the gear selectivity, for at least for the long lines. There's several things that can be tried to see if it's possible. Also, I mean, this species, the bycatch is it's some of the problem also, and in the past they were also fished by their oil. This has been reduced a bit. For the deep water sharks, at least the finning is not an issue, because usually they don't have big fins. So I guess maybe one of good approach will be trying selective or gear selectivity measures will be a possibility.
Yeah. Sophia, any offers, any bits of advice? How can a general member of the public get involved with the preservation of deep water sharks? We just have a few minutes.
Yes, I will wrap up. So the first question I think Raquel asked is that if it's possible to breed these animals in captivity for consumption. It's not. Still is not, because of the pressure. We would need pressurized tanks, and that would be crazy thing to do. There are a few species that were commercially used in the past, like the Portuguese dogfish and another Centrophorus, the gopher shark. But now, as Diana said, they are protected, so they must be returned to the water. So in order to help the deep water sharks and deep water, don't, let me say this political way.
Just say it, it's okay.
Yeah, no, I have to be careful. Okay, so I don't eat fish, I don't eat shrimp. So it's a way of trying to diminish my impact, and also avoid fisheries that are not selective. So bottom trawlers are not selective, right? They collect everything. Imagine a savanna with a huge net collecting zebras and giraffes and everything, but you just want to caught one species of leopard. I don't know.
Right.
You collect everything, you kill everything else. So this is not selective. It's not good. So first act, see what you're eating, the precedence, what you're eating, which fisheries, and try to avoid fisheries that are not selective. Bottom trawlers so far are not selective. So yeah, that would be
one of the things that I look for, excuse me, whenever I look for sustainable seafood is there's a lot of apps you can download. The Monterey Bay Aquarium has their Seafood Watch app, which is really great, and they actually get into how it was caught. Single line often is the best way. It's just a person off the back of the boat with a fishing pole catching fish, and that way they can be, as you were saying, selective in what they're catching. Is often one of the best ways. But yeah, do some research. Look out. A lot of grocery stores have like little labels on their fish and things, where it comes from.
Just a side note, Lance, bottom trawlers, they collect 70% or more of bycatch. So
Right.
from 10 animals, 3 are shrimp. Right.
The rest are something else.
The rest is dumped into the water, alive or dead.
Right.
So
Yeah.
it's,
So shrimp, not so good.
no.
Well, I think that you guys have both of you have really complementary research, and I think that could be an answer to it as well, is more research, right? More of you guys being out there in the field doing what you do, giving us more information about and more insight about these really important species, and how they have an impact on the ocean. And I know that your projects are still ongoing, so hopefully someone out there is listening and can see that it's worthwhile to fund these sort of things and keep them going and make them bigger and better. I know you both probably have a wish list a mile long that for what you would like to do, right? But next time we'll have you back to give us that list. I want to thank both of you so much for joining us this evening. It's been a real pleasure learning about deep water sharks and your research. It really sounds like you guys are amazing. I wish I could go out on one of your boats and do some and follow along and tag along. It looks really, really amazing. So thank you both for joining tonight.
Thank you very much.
Thank you very much, Lance. And
yeah, thank you. And for those of you listening, please, you can look up both of our researchers, Diana Catarino with the University of the Azores, Sophia and her Elasma project, Deepwater Elasmobranchs of Portugal project, and find out all about what they're doing and what they've been up to. For those of you that have stuck around, thank you so much. Next week is our last series of this year. Next week, I said next month, I should say, is the last series from our Distinguished Speaker Series for this year. We will have our speakers, two speakers again, Dawn Kroll and Melissa Cronin, talking to us all about manta rays, and be a discussion on the impacts of commercial fishing on manta rays, and what we can do to protect those really valuable species. So join us then, November 3rd. Oh, the date's wrong. Oops. It's the first Thursday of December from 6 p.m. to 7 p.m. Log in. You can register on the MODS website and join us for another really great discussion. Thank you so very much, and we will see you next time.
Thank you very much.
Thank you.
Bye-bye.
Bye.
Good night.